This is the second of a series of videos looking at the structure of DNA and how the chemistry of DNA can explain its role in allowing, for example, the copying of genetic material in living organisms. So in the first video we looked at the way in which the nucleotide monomers were formed from various subunits and how they joined together to produce a strand of DNA. In this video we look at how two strands are able to bond together to form a double helix structure through what's known as base pairing. So a reminder of what we've seen so far, the way that a nucleotide consisting of a sugar, a base and a phosphate can actually form a long strand by forming condensation reactions between the phosphate and sugars of the neighbouring nucleotide. What we've then noticed is the way that these bases stick out from the sugar-phosphate backbone at right angles. And that's sometimes thought of as being a little bit like a ladder, with the rungs of the ladder being the bases and the struts of the ladder being the sugar-phosphate backbone. So something like this, rungs and the struts of the ladder. Okay, so a reminder then that the nucleotide consists of a sugar phosphate bonded to a particular base. Here's the base that we call adenine, and here are some other nucleotides containing different bases: the base cytosine, the base thymine, and the base guanine. Again, those names are on the data sheet. You won't need to remember them, but you can, if you want in discussion, use letters A, T, C, and G if required. So here then is a single strand of DNA. Now we describe DNA as being a double-stranded molecule, so if that's one strand of DNA, what does the other one look like? Well, let's have a look. Here it is. And you should notice something really quite significant about the pattern of bases in this right-hand molecule. So pause the video while you think about that, and then we'll look at the significance of that feature. So yes, what you should notice is that when you've got an A here, opposite we've got a T, and then here's a T on the first strand. Opposite that we've got an A on the second strand. Similarly, C seems to be opposite G, and G seems to be opposite C. These pairs of bases are known as complementary bases, complementary base pairs, and you can sort of see that just the way they've drawn them here, they appear to fit together. Well, that's a kind of schematic drawing. What we're going to have a look at now is the way that actually that has some basis in the reality in terms of the structures of the bases. And the point is therefore that if we bring those two strands together, you can see how the bases indeed snugly fit into one another. It also illustrates why it is that it wouldn't really be possible for a base A here to bond to a C. The structures are not complementary to one another. So there's a kind of similarity there with the idea of active sites and substrates. Only one substrate will fit into a particular active site because only one substrate will fit and bind into that active site. Put a bit more detail onto that. So again, here we can see our base pairs, and this is starting to illustrate why it is that those bases are able to fit together. These lines here represent bonds between the bases. You might think from the fact they are drawn as solid lines that we're talking about covalent bonds, but they are in fact hydrogen bonds. So hydrogen bonds link the two bases together, and there they are. And notice that we're getting two hydrogen bonds between an A and a T, three between a G and a C. I think it's unlikely that you'd be required to remember that, but certainly we ought to be aware that there are multiple hydrogen bonds possible between these pairs of bases. So again, showing that in a little bit more colour here. Here again, a sequence of bases on one strand bonding to complementary bases on the other strand. Now, we said that hydrogen bonds are the things that bond the bases together. We can look in more detail now at the structures of those bases, and we can start to make sense of why those hydrogen bonds can form. Look at this pair of bases here, adenine and thymine. Look at the way that a nitrogen with a lone pair is able to line up to and form a hydrogen bond to this delta positive hydrogen here. And then, if those two form a bond in this orientation, look at how now the oxygen and hydrogen above it are also lined up in a way perfectly enabling them to form a second hydrogen bond. And that's even more significant in the case of these two bases here. Again, there's a hydrogen bond forming between a nitrogen and a hydrogen here, but look at how these two hydrogen bonds forming between oxygens and hydrogens are perfectly lined up to enable now 3 bonds to form between the guanine and the cytosine. And it's that lining up of multiple pairs of atoms that enables the bonding to be so perfect between these bases, and hence consequently why they always form that complementary base pair. So we are expected to remember the pairing of bases, adenine with thymine and cytosine with guanine. So, making up some kind of phrase to help you remember that would be certainly a good way of doing that. And we're also required to remember, obviously, that it's hydrogen bonds that cause that pairing. Now, just like you remember from your protein structure, the way that an amino acid chain often folds into the form of a helix, so indeed do these DNA strands. The phosphate sugar backbone tends to kind of spiral around, coil around to form this helix, and there are 2 of these backbones, and they're coiling around one another. Notice how the base pairs in the middle are actually relatively straight, relatively flat, and in fact it's interactions between those bases that probably mostly in the form of instantaneous dipole-induced dipole forces that help to stabilize that helix, amongst other features. So it is the hydrogen bonds between the bases that link those 2 strands together to form our double helix. More views of the double helix, slightly jazzy one here, and this second one here probably the way in which you're most likely to be presented with a diagram in an exam question if they decide to show you the entire structure. Now, this model of DNA is often described as the WatsonCrick model, after the 2 scientists that first proposed it, and we could be expected to remember the key details of it. And so here are those details. The model consists of 2 strands. The strands are arranged in the form of a double helix. Each strand is a sugar phosphate backbone attached to bases, and the strands are bonded together by hydrogen bonds between pairs of bases. So those 4 points there, which could obviously be scored by a labelled diagram, could well be the kind of thing that forms a 4 or 5 mark question in an A-level exam. Okay, so at this stage we're going to actually look at what we might be required to do in an examination question in terms of explaining how these hydrogen bonds form. So can you make sure you've got access to your data sheet and find the structures of the bases on the 3rd page of that data sheet. Here they are. Remember technically that these are not the bases in DNA because uracil is not present in DNA. Instead we have the base thymine, which has a CH3 group there, but it's easy to work out what that structure would be. Okay, so we're going to perhaps be faced with the kind of question you might have in an exam where you're asked to draw the hydrogen bonds that might form between cytosine and guanine. And so here are the 2 molecules. How might we be expected to do that? Well, this will attempt to illustrate how that might be possible in an examination question. So, start off with our molecule of cytosine as drawn in the data sheet. I think it'll be useful to remember that cytosine is bonded to the sugar molecule via this hydrogen atom here. Okay, and so that therefore connects up to the sugar phosphate backbone. So it's going to be this side of the molecule that's involved in forming hydrogen bonds. And we can probably suggest by looking at the structure of cytosine that this might be one of the molecules where 3 hydrogen bonds can form. Okay, let's see what happens. Okay, so we're going to bond that to a guanine molecule. So here's guanine as shown on the data sheet. Significant thing about that is that we know that this again is the atom that's used to bond to the sugar molecule. So therefore the kind of atoms that might be involved in forming hydrogen bonds again are likely to be found in this region of the molecule here. That's a slight problem because they're actually facing away from the cytosine that we've drawn over here. So you may well have to flip the molecule round a little bit. My suspicion is that probably the examiners will do that for you, will probably give you maybe the structure of guanine in a kind of helpful way, but I'll just suggest what might happen. They may flip it over. We've actually kind of produced a sort of mirror image of it here, and then we've kind of taken that molecule and moved it around a little bit. So we've taken this region here of the molecule and bent it towards the cytosine. And now you can see that when we do that, groups on our guanine molecule are in the right kind of orientation to form those 3 hydrogen bonds. So we've got a delta positive hydrogen on our NH2, and that can bond to a delta negative oxygen there. We can get the delta positive hydrogen on this nitrogen to bond to the nitrogen on the cytosine, and finally the delta positive hydrogen on that NH2 might be able to bond to the cytosine there. So that's the kind of pattern of hydrogen bonds that might form. You wouldn't have to generate this from scratch. Almost certainly what would happen is they would give you partial structures, and you'd need to really kind of think about rotating the structures in the data sheet to actually figure out which atoms go where. And so that will be the kind of thing we would end up with. Here's a diagram of it showing the hydrogen atoms in a bit more detail, and you can see here the way those 3 hydrogen bonds are formed in just the right kind of orientation. And the final thing is very much a kind of biological idea. Why is that hydrogen bonding and base pairing significant? Well, because it relates to the idea of replication. That means copying of genetic information. What we know is that living things can reproduce themselves. In order to do so, they have to make copies of their DNA. And Watson and Crick were able to use their model to explain how that might happen. So very briefly, without going into biological detail, here's what goes on. So we've got our DNA molecule here, and what happens to make a copy of DNA is that enzymes start to break apart the DNA, pull apart the bases to create kind of exposed bases here. Okay, that happens via an enzyme. Need to worry about what the enzyme will actually be called. My feeling is it isn't actually called DNA polymerase. It should in fact be called helicase, but we're not required to remember that detail there. What actually happens now is that having exposed the bases, nucleotides that exist in the nucleus are able to float in and form bonds to the exposed bases on these strands. Let's see how that might happen. Here come the nucleotides. Okay, what you'll notice here is that we are kind of forming now a new strand on one of our original strands, and that new strand looks very much like the original DNA strand that used to be bonded to this first one. In other words, what we are starting to do now is making 2 identical copies of our original DNA. We're not quite out of the woods yet. All we've done at the moment is to stick on these nucleotides. And so this is now where the nucleotides bond together, and this in fact is where the enzyme DNA polymerase actually joins together those nucleotides to produce 2 completely identical DNA molecules. And so we've now copied that genetic information, and that's all happened because of the fact that these bases are able to bond together and bond together in very specific pairs. So each DNA molecule consists of 1 original strand and then 1 strand that has been formed from 3 nucleotides but is an exact copy of the second strand that used to exist on the original DNA molecule. And that's the end of the second video looking at DNA structure. In the third video, you'll be able to see in the way in which DNA directs the synthesis of proteins.